Hello viewers, welcome to my channel. Today we will discuss about the identification of some microorganism based on their colony characteristics when grow on different culture media. In one of my previous video, we have already discussed about different kinds of culture media such as selective media, differential media, enrichment media, basal media, etc. Some media distinguish among different groups of microbes and even permit identification of microbe based on their biological characteristics, whereas some culture media encourage the growth of fastidious microorganism. Even some media select for the growth of particular selected microorganism. So let's start about the identification of different microorganism based on their colony characteristics when grow on different culture media. The first one is Eosin methylene blue, that is EMB. It is used for the identification of E. coli, that is Escherichia coli. E. coli colonies have a green metallic sheen when growing on EMB, that is Eosin methylene blue media. Next one is the Macconky Agar. It is also used for the identification of E. coli. In this media, that is Macconky Agar, the E. coli colony appears red. Third one is the Mannitol salt agar. Mannitol salt agar is used for the identification of Staphylococcus aureus and also identify the coagulase negative Staphylococci. Staphylococcus aureus produce yellow colonies with yellow zones, whereas the coagulase negative Staphylococci produce small red colonies with no color change in the media. So the Mannitol salt agar is used for the identification of Staphylococcus aureus and also the coagulase negative Staphylococci. Next is the TCBS, that is Thiosulfate bile salt sucrose agar. It is very important. This media is used for the identification of Vibrio species in a specimen. The sucrose fermenting Vibrio species such as Vibrio cholerae produce large yellow colonies, whereas the non-sucrose fermenter such as Vibrio parahaemolyticus produce blue to green center colonies. So TCBS mediums, that is thiosulfate bile salt sucrose agar, is used for identification of the causative microorganism for cholera. Next one is the Sorbitol MacConkey agar. It is a differential media. It is used for the identification of E.coli O157:87. It is an enterohemorrhagic E. coli. It produces a toxin, that is Shigella toxin, like toxin. The E.coli O157:87 appear colorless colonies. So in SMAC, that is Sorbitol MacConkey agar, the E.coli O157:87 appear as colorless colonies. Next one is the Hektoen enteric agar. Short form is HE. It is used for the identification of Salmonella and Shigella. Salmonella produce the transparent green or blue-green with or without black center and appear as almost completely dark colonies, whereas Shigella produce green transparent colonies. Next is Loeffler's serum slope. In short, it is known as LSS media. LSS is very important. It is used for the identification of Corynebacterium diptheria, which is the causative microorganism of diptheria. The colony morphology of Corynebacterium diptheria in Loeffler's serum slope is minute and cream colored. Note that the colony of Corynebacterium diphtheria in LSS media is minute and cream colored, and the metachromatic granules seen in methylene blue stain or Albert stain. Metachromatic granules is another important characteristic for Corynebacterium diphtheria. Another important thing about this LSS is that this media is not sterilized in autoclave. As the serum containing media is highly susceptible to high temperature, so it should not be sterilized in autoclave. It is sterilized by inspection. Next one is LJ medium, that is Lownstein Jensen medium. This media is used for the identification of Mycobacterium tuberculosis, the causative microorganism for tuberculosis. Mycobacterium tuberculosis appear as a brown granular colonies, sometimes also called as buff tough rough, when grow on LJ medium. The last one is Deoxycholate citrate agar. It is used for the identification of Salmonella strains that cause food poisoning, and also Salmonella paratyphi, that is responsible for paratyphoid fever, but it is not so selective for the identification of Salmonella typhi. It produce, that is Salmonella produce yellow or colorless, often with black center. So this is all about the identification of different microorganisms based on their colony characteristics when they grow on different culture media. So thank you. Please like, share and subscribe to this channel.